This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 194, recorded on February 7th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, how are you?
Hello.
Are you doing all right?
I am, and you?
The weather. I'm fine. I'm in California, so Southern California. I'm doing fine.
Well, it's weird here. It gets cold and it gets warm and cold, and today it's warm again. Yesterday it was raining. Last week it was freezing.
We had a fair amount of rain and a lot of mushrooms.
Yeah. All right.
So life makes sense again.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. And I'll add that here in Michigan, the weather is rocketing between ice and like today it's going to get to be 50 degrees, but not until like 7 o'clock at night.
It's weird.
It's just weird. Yeah.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Michael's teaching now, right?
Oh, yes, just as you are. You're doing virology and I'm doing medical microbiology.
And Michele, you teaching?
Not for a while. I have the medical students, but not until late May, early June.
So I'm teaching.
Yeah, what are you teaching?
Well, I help run a graduate seminar for first-year graduate students. It's called Integrative Microbiology.
And that's at San Diego State?
No, it's
UCSD?
At UCSD with students from San Diego State as well as.
Oh, very good.
It's a lot of fun.
Well, I don't know if listeners know this, but I record all my virology lectures. You can find them on the web. You can go to youtube.com/PROFVRR, or you can go to virology.ws/course. And we are at lecture number 5. And every year I put new videos up. So if you want to learn about viruses, start there.
The latest.
The latest on viruses. That's right. Everything I know.
And I'll add that if you follow Vincent on Twitter, you would have learned that.
That's right.
You posted that information to your large following.
I try to tell people.
Public service.
I try to do that. I love to teach and give out information, and that's part of what we do here on TWiM, of course. It's all about that as well. Now, today we have a snippet and a paper, and after we're done, we're going to give away a book.
Ooh.
I have another book to give away. And so listen, because you never know when it's going to pop up. And I would like to start with a snippet today, an article from Cell Host and Microbe. It's called A Gut Commensal Bacterium Promotes Mosquito Permissiveness to Arboviruses. And of course, arboviruses are arthropod-borne viruses. And today, the mosquito is the subject. And this comes out of multiple institutions in China's Tsinghua University, the Shenzhen Center for Disease Control and Prevention. It's a state key laboratory in Beijing, the Beijing Institute of Microbiology and Epidemiology, Southern Medical University, Yunnan Institute of Parasitic Diseases, and then the University of Connecticut in Farmington. Rather mundane, I guess, compared to the others. The first author is Pa Wu, and the last author is Gong Chen. Now, just like us, mosquitoes have a gut microbiome, and we are learning slowly what this microbiome does. And in mammals, we are getting interesting information that the microbiome in the gut can modulate infection by viruses that infect the gut. For example, in mice, poliovirus doesn't infect very well unless there are bacteria in the gut. If you give mice antibiotics to reduce bacteria in the gut, the virus doesn't grow well. And reovirus is another virus that shows similar properties. And there are a couple of examples of that. And so today, the question is, what about the mosquito gut. And we know already from a study we actually did on TWiM earlier last year, there is a fungus in the gut of mosquitoes, in particular Aedes aegypti mosquitoes, that if you take it away, dengue virus doesn't replicate very well. So it increases the replication of dengue in the gut. And what it does, it appears to secrete something that inhibits digestive enzymes in the gut, which would otherwise destroy the virus.
All right. That's neat.
That's pretty neat. And today we're going to see about the bacteria in the gut.
And their role, and whether or not they're as important as the fungi.
And the virus, again, is dengue. And the mosquitoes are Aedes aegypti. And they use a laboratory strain called the Rockefeller strain, which is extremely wealthy. By the way, that's
an inside joke.
It's a very wealthy mosquito.
Mosquito.
And you can give these mosquitoes a blood meal. You can give them blood laced with dengue virus, and the virus goes into their gut tract, and it will replicate in the epithelial cells lining the gut. Then it gets into the circulatory system of the mosquito, the hemocoel, and then it spreads systemically. And then if it's going to be transmitted by the mosquito, it has to reach the salivary glands, the virus that is, and then it will be transmitted.
When it bites you.
When it bites you again. So these are female mosquitoes who are seeking blood in order to make eggs. They need the blood for that. Male mosquitoes do not drink blood. They just drink sap of plants. And so if there are viruses, if the host the mosquito is initially biting is viremic, it picks up viruses and it can redeliver them. So they're asking in this paper, what is the effect of gut bacteria? So when they feed these mosquitoes antibiotics to decrease the bacteria in the gut, it decreases dengue virus replication. You get tenfold less dengue virus.
It's kind of amusing, isn't it? Sort of a case where antibacterial antibiotics help in a viral infection.
That's right. That's absolutely right.
But only in the mosquito. So an antibiotic won't help you as an infected individual should you have dengue. So don't go asking your physician to write a script for an antimicrobial if you got dengue, because you're not a mosquito, folks.
No, not mosquitoes. Okay, so then they said, what bacteria is the culprit? And so they cultured the bacteria from the gut using broths, and they were able to culture 21 different bacterial species from the midgut of these Rockefeller mosquitoes. And so they're only looking at the culturable ones, but these are the ones that they could do. And then they grew up each of these 21 bacteria, and they fed them to mosquitoes along with dengue virus. And they asked which one, after antibiotic treatment, they asked which one will restore the ability of dengue virus to replicate in mosquitoes. And I would have said this was never going to work because you probably need more than one, or maybe the non-culturable one is the one that does it. But it worked.
but you know, Vincent, this seems to be becoming a common thread. We had that paper of a few weeks ago where we were discussing the lactobacilli one lactobacillus being able to alleviate the symptoms of autism spectrum disorder. And here again, we have one microbe that's going to be involved in determining whether or not the dengue that the mosquito has picked up from the infected blood meal is able to replicate and continue the infectious cycle. So it's really interesting how the microbiome is helping us with limited neurons appreciate what's going on.
Well, I think the moral here is that just do the experiment. Don't overthink
be biased.
Don't overthink
Don't
it.
Biased.
Because if your advisor may say it's never going to work, but just try it.
From your lips to the study section's ears.
And so, yeah, right. So the bacterium involved is Seratia Marcescens, which some of you know for a long time. I remember my first encounter with Serratia on a plate. It was interesting because it was pink. And so that's
it's responsible for putting a red ring around the toilets of Charleston.
Is that right?
Yeah, it grows in toilet water in Charleston, and if you don't routinely clean your bowls, you'll actually get a red ring from the Serratia, because toilet water is generally about room temperature, which is ideal for it to make pigment at.
All right. Well, so Seratia Marcescens from the mosquito gut is the bacterium that can help dengue virus to replicate. So how does it do that? Well, another interesting series of experiments. They grow it up in serum-free medium, and they take the supernatant. They figure maybe it's something secreted from the bacterium. So they take the supernatant. They actually test both the lysate and the supernatant. And this is a supernatant when fed to mosquitoes that enhances dengue virus replication. And then eventually they do some fractionation and mass spectrometry, and they test individual proteins one by one, just like they tested the bacteria. And they identify a single protein, a peptidase called enhancin, which enhances dengue infection of the mosquitoes. So they name it smenhancin, Seratia Marcescens enhancin. And that seems to be the major molecule that's responsible for this. If they make antibodies against smenhancin and these block enhancement of dengue virus, they knock out the smenhancin gene from Seratia Marcescens, and they show this bacterium that does not make smenhancin, does not enhance dengue virus infection. And it also works with some other arboviruses, Zika virus and Synbis viruses. So a single...
those are unrelated, aren't they?
Synbis and... Synbis and Zika. So Zika and dengue are both flaviviruses, but
right.
Synbis is an alpha virus, a different family, and it's vectored by a different mosquito. It's actually vectored by culicine mosquitoes, and it turns out that the enhancement by Marcescens doesn't happen in its culicine mosquitoes. If you feed them S. Marcescens, it doesn't make any difference with that virus. It only works in Aedes mosquitoes. All right, so what's happening? What is the smenhancin doing? Well, I'll cut a long story short here. It turns out that this is an amazing amount of work also. The hypothesis is that smenhancin is digesting the glycoprotein layer on the gut epithelium. It's called the mucin layer. So the epithelium of your gut is covered with sort of mucus, and they're thinking that's blocking virus from getting in effectively. And the smenhancin must be getting rid of it. So they look in the Aedes aegypti genome. They find 12 genes that encode mucin-like proteins. They find 6 of them highly expressed in the mosquito gut. They individually silence them one by one with RNA, and they identify 2 that are involved in enhancement of infection. So when they knock them down, you get enhancement without having to give the smenhancin-producing Serratia. And the idea is that these proteins are called amucins. They're substrates of smenhancin. And they actually show that if you produce the proteins in other cells, they can be totally digested by smenhancin. It's really remarkable. It's all working out perfectly here.
Yeah.
And in fact,
clockwork.
they even look in the mosquito. They give the mosquitoes the bacteria, they section them, they look in the gut, and you can see the mucin layer is gone. It's almost eliminated in the gut treated with smenhancin. It's really remarkable. And then they do a couple of other experiments which are quite interesting. There are other bacteria that make enhancin-like proteins that are gut commensals in mosquitoes, like Bacillus species, Enterobacter, Enterococcus, Klebsiella. So they cloned out the enhancin genes from all of them. They gave the proteins, they expressed the proteins, gave them to mosquitoes, but none of them enhanced dengue infectivity. Only Serratia is the one that does this. It's quite interesting. And then the question, of course, is what about in the field? This is a Rockefeller mosquito, but what about field mosquitoes? So they go out in the field in different areas of China, and they collect a bunch of wild mosquitoes, Aedes aegypti mosquitoes, and three of them have Seratia Marcescens in their gut tract, but the others do not. And the 3 that do not, when they feed them Serratia, it enhances dengue replication in them. So they are thinking that out there in the field, maybe, the presence or absence of Seratia Marcescens can control dengue prevalence.
And it really probably depends on how the mosquito's gut actually picks up the Seratia.
Yeah.
Because whatever the local environment may be that the mosquitoes are developing in, the pond water, if you will, it may be absent the Seratia Marcescens, and consequently dengue, which is not prevalent in that particular region of China, isn't, if you will, endemic in China simply because of the absence of Seratia as the mosquito is developing, so the gut isn't being seeded.
Now, we have Seratia in our guts, right?
Yeah, it's transient, hence the toilet water issue.
So it's not there all the time?
Well, it depends on the individual and what you're colonized with and whatnot. It's a normal flora organism that is generally not a pathogen of humans.
Yeah. Now, of course,
that's encouraging, because if we wanted to do some kind of application out in the field with Seratia, we wouldn't need to worry so much about risk to humans.
Yeah, that is indeed the case, but it can cause pneumonia because that was, of course, the very famous 1950s where the accidental invention was the sonicator, because they were sonicating bacteria into the Bay Area of California, and the microbe that they were sonicating out, looking for its presence in the San Francisco Bay Area, was a particularly pigmented variant of Seratia Marcescens, and that's how they learned that sonication, which was designed to aerosolize the Seratia out of the back end of the airplane. That's when they figured how many kilocycles of sound were bactericidal.
So they did that to look at dispersal patterns,
yes,
right? And they figured, Seratia is not a pathogen, so we can use it, but it turned out to make some people sick, right?
There's some people who were immunocompromised or at higher risk for developing pneumonia, typically the elderly or the very young, develop Seratia-based pneumonia.
And again, that required intervention by people, engineered dispersal, not natural dispersal.
Yeah.
Not natural dispersal, absolutely.
Right.
And remember, everyone, that dengue in humans does not infect your gut. It's delivered by the mosquito into your blood, and it circulates and infects various cells in different tissues, but not the gut. It's not a gut pathogen. So Seratia is not going to do anything in humans for dengue. But of course, it's the interesting thing about these viruses that go between mammals and mosquitoes. You have two very different hosts that you have to deal with. So I thought these data were very convincing. I am really surprised that it's so simple, that it's one bacterium and it's one protein. So I wonder if there are other contributions in there, in the non-culturable bacteria, for example. I just don't know. And
it certainly is worthwhile hypothesis.
And the other part is, now the presence of Seratia in the mosquito gut, it's not there for dengue virus, obviously. It's doing something for Aedes aegypti. And if you look at their experiment, this Seratia Marcescens completely denudes the gut epithelium of mucin. And I can't imagine that that's something good for the mosquito, right?
Are they feeding off? Are they feeding off the carbohydrate that they liberate?
Unless it enables the blood meal to more effectively transfer nutrients to the female so she can develop more eggs.
So maybe it's induced and it's not always produced. That could be, right?
Yeah.
Yeah, because I'm thinking if it's always denuding the epithelium, that might not be good. On the other hand, they were feeding the mosquitoes the purified protein, so maybe that's not the way it normally is, right?
Yeah.
Just the bacteria there probably remove bits here and there for whatever reason, maybe to enhance absorption or something. But, it's not in every mosquito isolated, so it's not required for all of them. I don't know. I don't know if it's required for any of them, actually. We just don't know, right?
And it was just for this one particular virus. As you said, it didn't work for Sinbis virus. And, the virus is, again, a parasite of the mosquito. If you will, the mosquito is the accidental host for the virus in that particular sense.
The virus doesn't seem to harm the mosquito either.
No.
They seem to be okay with it because mosquitoes don't live very long, about 2 weeks. But they seem to be able to take a blood meal, have the virus replicate, and then take another blood meal. Of course, they have to take at least 2 in order to deliver it, right?
To transmit.
To transmit it. So they have to be healthy enough to do that. So the viruses don't hurt them.
But the only thing I was looking for in the discussion as to their comment as to whether or not it would change the clutch size of the eggs that the mosquito would ultimately lay if it was infected with dengue. And that may be already known. And I didn't have time to ferret that out.
Yeah, I don't know. I think that's been looked at. I don't remember the answer, though.
Yeah.
Now, one thing they do say, they said 2 things I want to point out in closing here. First, they said maybe we could control viruses by somehow controlling Seratia in the mosquito population. And they say either through Seratia-specific phages or bactericidal chemicals in dengue endemic environments. And I was wondering what you thought of that, the idea that you might control the virus by controlling the bacteria.
My answer to that is there are cheaper methods to control mosquito spread than trying to disrupt an entire ecosystem of one microbe.
Right.
I don't want to see mass release of an engineered Serratia into the greater pond system of China. Mosquito nettings would limit viral spread. And remember, the mosquito has to get the virus from an infected host, namely a human.
Mm-hmm. Mm-hmm.
Because the human is the natural reservoir is one of the reservoirs for dengue. I don't know if it is the natural reservoir for dengue.
It's the most significant. There are monkey reservoirs, but with the big human populations, that's enough to sustain transmission.
Yeah, it's enough.
Yeah. I agree. And also we know that we can engineer the mosquitoes. We can put Wolbachia bacteria into mosquitoes, Aedes aegypti, and that restricts dengue replication. It's been a wonderful study done in Australia, where they did a release of Wolbachia infected Aedes aegypti, and it eliminated dengue in this small town over the past 4 years. So it's really remarkable. And the other point I wanted to comment on, they say enhancin-like genes are encoded by many bacteria in humans and in mice, and so maybe enhancins regulate infection of enteric viruses in mammals, and that's an interesting point. It would be interesting to look at that, I think, in a mouse model. I think that's
there is precedent for that type of interaction at a different mucosal surface. Jeff Weiser's group has looked at the respiratory tract and found that colonization by Streptococcus pneumoniae, which is a bug that makes an enzyme that cleaves sialic acid, actually increases susceptibility to flu virus. So there are these interactions. If there's a microbe that changes the carbohydrate surface of the respiratory epithelium or gut epithelium, you can then impact viruses, either positively or negatively, ability to bind.
What about polio, Vincent, and Peyer's patches? Is there anything known about polio infectivity in Peyer's patches and whether or not there's any of this mucin-like interaction with the gut flora?
So not mucins, in the case of polio, you do need bacteria to allow infection in mice anyway. And one of the components at least is LPS. And it seems to stabilize the virus particles. Right?
Okay.
So without it, the virus particles seem to fall apart in the intestine and lose infectivity, and they're stabilized by it. But that's only in mice. We don't know if the same thing happens in humans. But I think the enhancin question is interesting, whether that plays a role in giving access to epithelial cells, just like here in the mosquito. That would be interesting to look at. You can imagine some experiments in mice looking at that. So hopefully this will stimulate that. Anyway, I thought that was a cool paper, and I thought TWiM would be the right place because it's mostly, but it could be a crossover, right? It could be either TWiM or TWiV, but TWiM got it.
TWiM got it.
All right. Now, before we move to Elio's paper, I want to give away a book, and it is a book published by ASM Press, and it's an edited book by Robert Britton and Patrice Coney. It's called Bugs as Drugs, subtitled Therapeutic Microbes for the Prevention and Treatment of Disease. And it was just published last year. And and as you would guess by the subtitle, it's all about using bacteria for preventing and treatment of various diseases. We have a section on, and each article is written by different authors, section on traditional probiotic approaches, next generation bacteriotherapy we have for colorectal cancer, nutritional disorder, liver diseases, osteoporosis. Then we have a chapter on control of infectious diseases by microbes, C. diff, enterococci, next generation microbial therapeutics, and indirect strategies to target microbiome function. So really interesting topics, and this is yours to have, and all you need to do is send an email to twim@microbe.tv. The subject line should be Bugs as Drugs, and we'll pick one out of the.
And I guess the participants in this podcast are not qualified to submit their name.
Yeah, that's so funny.
I'd love to get this book.
It's a wonderful book, and you can enter, Elio. It's no problem. You have to send an email to twim@microbe.tv.
I'll buy it. I'll buy it.
And it's a wonderful book, yeah. And I got it last year from ASM Press. And so, yes, twim@microbe.tv, we'll pick out a listener. Now, you don't have to write anything in your email. However, we love to hear from you, so we won't object if you do write to us. All right, Elio, what do you have this week?
I have a paper called Minicells as a damage disposal mechanism in Escherichia coli. The authors are Camilla Rang, Audrey Proenza, Kristen Butz, Chao Shi, and Lin Chao, all from University of California, San Diego. These are people some of them I know personally. It's an interesting paper because it deals with an old question. About 50, 60 years ago, a guy named Howard Adler discovered that bacteria which were resistant to radiation, E. coli, resistant to radiation, made not only its normal cells, but also it budded off little spherical bodies, which he called minicells. They are the size of the diameter of a cell, but not the length of the cell. And what was interesting about them is that they contained no DNA. They had the cytoplasmic content, that is ribosomes and proteins and so forth. And what was interesting about it is that they were very good for manipulating a lot of things. Like, for instance, if you had a bug with plasmids in it, plasmids are extra chromosomal genetic elements, usually circular. These would segregate into the minicells. So the minicells would contain no bacterial DNA, but they contain plasmid DNA, which means that in the old days, if you want to study what is it that plasmids do, for instance, what proteins do they synthesize, this was a godsend. This was the way to study it because you could separate the bacterial DNA from the plasmid DNA. Am I clear about that?
Mm-hmm, mm-hmm.
So minicells were really been around. Now, normal cultures make practically no minicells. If you can find them if you know how to look for something that happens once in 100,000 or 1,000,000 times. Very rare. But the mutants make them all the time. So the study of minicells became interesting. They've been around forever. And as I say, there's a renewed interest because they can be used today as little packets of cytoplasm that carry drugs, for instance, anti-cancer drugs. You could make minicells fill them up with cancer drugs in one way or another, and this would be a nice delivery system.
Because they would be pinocytosed or endocytosed by the eukaryotic cell.
That's right, exactly. So there's still a lot of interest, but the basic question that the authors are actually addressing is, what are they for? because obviously E. coli is not in the business of making something which is useful for cancer therapy. So their view is the following. Bacteria often make misshapen, misfolded proteins, which aggregate thanks to chaperone proteins. They aggregate into little balls. These little balls are bad stuff. They're not useful. They're misshapen, as I say. They're non-functional proteins. And a cell would do well to get rid of them. Okay? Now, what comes to mind? Put the two things together, and what they come up with, as the title tells you, the title again is Minicells as Damage Disposal Mechanism in Escherichia coli. So they propose that the reason cells make minicells under some conditions is that they want to dispose of unwanted proteins. Okay? It's a garbage disposal handling mechanism. The idea is novel. The idea is good. Not without a little, maybe a couple of maybes that I'll toss in a little bit later. So what do they do? Well, the experiments are really very elegant. They're very nicely done. They use streptomycin at a low concentration, 6 micrograms per ml, which is enough to kill or to, eventually to kill or to keep normal E. coli from growth. But these mutants, which are called mutants that make minicells, are resistant to streptomycin. So what gives? Well, streptomycin at that concentration induces the formation of unwanted proteins, just the kind of stuff you want to get rid of. And so they do that. So it happens that when you add streptomycin, you make more minicells. The number of minicells made goes up, which is like in keeping with the prediction. You add streptomycin, you make unwanted proteins, you get rid of unwanted proteins by making more minicells. It sort of fits, doesn't it?
Tantalizing result.
It's a neat result, really. I don't know that I knew that. I think that streptomycin has been studied extensively, but these low, almost subbacterial static concentrations, not many people have looked at it. 6 micrograms of streptomycin is not 100 micrograms of streptomycin. So anyhow.
But Elio, I'll bet you do remember this, because in 1960, Bernie Davis published in Nature the effect of streptomycin on E. coli. And it's a really telling, albeit very short paper, where the telling line in the paper that you're going to then expose us to as you take us through the data is that when these cells are exposed to streptomycin, at 45 minutes, what happens is the cell begins to excrete nucleotides from the cytoplasm, suggesting that the E. coli has lost the integrity of its membrane or its permeability membrane. And so as you begin to reveal the rest of the story, I encourage the audience to just remember that what the streptomycin is doing, which is heretical because we all think of streptomycin as inhibiting protein synthesis. But in addition, streptomycin alters the permeability of the cell, which is principally the cytoplasmic membrane, which is going to result in oxidative damage to the cell, which will then oxidatively damage the proteins. So, Elio, now you can go back to revealing the rest of this wonderful story.
Well, one of the things that is most interesting is that when you look at the location of the misfolded protein, as I say, they make little balls, aggregates called inclusion bodies. And where do they find them? You find them at the poles. Now let's go back to the business, the minicell formation business. This has been studied in beautiful detail, and there's a lovely story behind it, because what happens is that the reason mutants make mini cells and normal cells practically do not is because they have inhibited division at the poles. Imagine a hot dog. That's E. coli, long E. coli. Imagine that it normally divides in the middle, but it could divide any place else as well, right? You take a knife to it and you can cut it any way you want, but you only cut it in the middle. However, the ends, if it cuts at the ends, you would get a mini cell, wouldn't you?
Mm-hmm.
Not at the very end, but before the curvature happens, at the end of the sausage. If you cut a little bit before that and you daub a little bit of mustard on it, you would find the mini cell has been labeled. Okay. Now, why does this not happen? It does not happen because there's a beautiful system of 3 proteins called, not surprisingly, min A, min B, min C. Actually, it's more like min D and min C and min E. C, D, and E. They actually, I don't remember what happened to A and B, but it's C, D, and E were actually described by Howard Adler. They work this way. Min C is an inhibitor of a system which exists in bacteria for cell division. This is a system that depends on forming a ring around the periphery of the cell, which constricts. In other words, as it constricts, it makes the cytoplasm smaller and smaller until it divides. This is how bacteria divide, and this is called the FTSZ, Z if you're British, system. It was discovered by Lichtenhaus, man at the University of Kansas. A beautiful system which is universal. Practically all bacteria and even some chloroplasts have it, and this is how they divide. So if you want to divide, you make an FTSZ ring. As I say, by the way, the FTSZ is a tubulin-like protein, which is a structural protein, and it has this magic ability of constricting, of making a constricting ring. So why it is not made at the poles, and the reason it's not made at the poles is because min C in an active form, the story is a bit more complicated, I'll simplify it, but min C at the poles is what inhibits the formation of the FTSZ ring there. Okay? So now we know why normal cells don't make mini cells and why mutants do, because a mutation in in min C will allow the formation on an FTSZ ring at the poles. You with me?
Got it. Yep.
Okay. Now, they look at it in some detail, and they find that the mini cells are not made at both ends of an E. coli. Now, think about an E. coli again in terms of how it came to be. Here's a little sausage that came to be because its mother divided into 2, right? You take a sausage, you take a knife, and you cut it in half. You have 2 daughter sausages. Okay? Now, when you cut a sausage in 2, you create 2 new poles, 2 new ends, right? The cut piece. Let's call those the new ends. Let's call the other ends, which were there before, the old pole. So it turns out that these 2 are not identical. Of course, the new pole in the next division is going to become what? It's going to become the new old pole. Sorry if I make this a little bit more complicated than it needs to be. So new poles are the ones that are made at the new division. Old poles are the ones that were made at the previous division. And where are mini cells made? Mini cells are made only at the old poles. And guess where you find the inclusion bodies, those little balls of unwanted protein? You guessed it. You find them at the old poles only.
Aha.
Aha is right. The plot thickens. So now you say, maybe what's happening is exactly what you had set out to find, namely that the production of mini cells is a device for making a garbage disposal mechanism. Inclusion bodies are made under stress, such as a small amount of streptomycin or radiation or anything you want. Proteins are made in substantial amounts that are not functional, and the cell wants to get rid of them because they are in the way. Imagine, for instance, if you made some of these unwanted proteins in sufficient amounts and you didn't package them in inclusion bodies, but they were all over the cytoplasm, they would interfere mightily with metabolism. So you really want to get rid of them. They would be in the way of transcription, translation, who knows what all. They would do bad things. So you want to get rid of them, you package them, you put them at the ends of the cell. Now, at the end of the cell, they sit there and are inert, but you still want to get rid of them. And so you invent the mini cell mechanism to make for a garbage disposal of that sort. Now, they go into a fair amount of detail about this, and it's an elegant paper, I think, in many ways. One of the things that's kind of fun is they measure what good does it do to the actual cell itself, not to the population, but just to the cell itself that makes mini cells and so forth. So they found that making mini cells, as you would expect, is at a cost. They amount to about 20% the volume of a young E. coli. So there's quite a bit of material there. There's quite a bit of protein and nucleic acid and lipids and so forth. So that takes time. And sure enough, if you measure the time it takes for the cells to grow, cells that are making and in the process of making a mini cell grow a little bit more slowly. However, the next generation, that is the cell which comes off one that made a mini cell, the next daughter cells. Imagine you made a mini cell, now you divide. You call those daughter cells. The daughter cell from the end that had the mini cell grows faster. Okay, so it has an advantage. So you rid this unwanted garbage and you grow faster. And that's pretty much the story, I think.
And can I just interject? They do a beautiful job of providing schematics of these hot dogs with the terms, and they've color-coded each of their schematics. So then you can look at their bar graphs.
That's the mustard.
Yeah.
They're decorating the sausage.
Yeah, see the difference in the populations, including the doubling time, as Elio just mentioned. So it's really very easy to follow.
That's right. There's lots of beautiful pictures because they use a lot of fluorescent in various ways, all of which are very elegant. So that's right. They go on to study do some population studies, which I don't think I need to get into, but they say that in mixed cultures, a strain lacking MinC, that is, a MinC mutant, actually does better in cultures. And this is complicated, but there's no reason for going into it at this point. So the message is pretty much that. You make mini cells. Why? Not just because you want to divide an extra time and make a lop off a little end from the sausage, but because you want to get rid of unwanted proteins which are in the way of the metabolism of the cell. I think, in truth, this is the conclusion is a little bit still up in the air. I mean, there's a lot more work that needs to be done. I think this is an elegant beginning, and I'm sure that they're going to continue working on this field. But it is very suggestive and I think believable. I mean, there's nothing that is in the way of thinking that this is one of the reasons why bacteria make mini cells.
I agree. I found one of their really beautiful, convincing figures was figure 7b, where they did an experiment and just made a 1-to-1 ratio of wild type and MinC mutant cells, and then exposed them to either no streptomycin or 2, 4, 8, or 16 micrograms per mil of streptomycin. And then 24 hours later, they asked what was the ratio. Was it still a 1-to-1 ratio? And what they found is in the absence of streptomycin, the wild type became predominant. It had a growth advantage. But at 4 micrograms per mil or up to 16 micrograms per mil, we see that the MinC now takes over the culture. It's growing, has a growth advantage, which is consistent with what Elio said. They're able to dump their oxidized clumps of proteins and dump them out their old pole, and then the daughter is free to replicate more efficiently.
But isn't the strep inhibiting protein synthesis? So why would the MinC mutant be able to replicate at all?
It goes back to the concentration of strep that they're using.
They're using very little.
I see.
At 6 mics per mil,
I see.
that generates more damaged proteins by inducing the mistranslations that streptomycin is known for.
Got it. And
that was the work of Davis back in that Nature paper in 1960.
Okay.
And Davis is commenting that it makes the cells more sensitive to oxidative damage, which in a sense is creating the garbage that necessitates or selects for the development of a garbage disposal. So as Elio said, we don't muck up the rest of the cytoplasm with all this trash lying about so that you can get the efficiency.
One of the points that they make, which is, I think, cute, notice that this is, in a sense, a way of making cells resistant to an antibiotic.
Yes.
Okay. And they say maybe this is worth looking at. Maybe that in an era where resistance to antibiotics is such an important central subject, maybe one should think of this as yet another mechanism on how bacteria can become resistant to antibiotics. And that, by taking advantage of the fact that minicells get rid of unwanted proteins, which are made during antibiotic treatment at the right concentration. Let's say it's worth looking at.
Yes, certainly for antibiotics that cause oxidative damage and would denature proteins.
There are people who believe that all antibiotics do, one way or another.
I see.
What other kinds of stress would induce mini-cell production? They only look at streptomycin, but have others?
Any strong oxidants, ultraviolet radiation, gamma rays, and in fact, that was known for a long time of how they got mini cells. You shine a little UV light on them to effectively give the cell an opportunity to make the mini cell.
So if you used gamma rays or UV, you would predict the min mutants are going to be more resistant, right?
Yes.
Would have been nice to have a different stress in here, I think, don't you think?
That's right. I think that's correct. And I think they will probably follow up on their own advice.
Yeah.
Let me read what it says. The generality of the effect of mini cells in response to streptomycin needs also to be examined for other antibiotics. If the response is general, our results could be uncovering a new mechanism that is used by bacteria to resist antibiotics. Amen. Well put. So that's the story.
You know what's I found really interesting here in the very beginning of the results, they write the description of materials and methods is abbreviated in this section to not overload the presentation of the results. Thank you. That's great.
I want to be very, very thankful for that. I think the paper is really very well written.
And to your point that they could have tried other stressors, it gives me an opportunity to share that all the data collection was done by eye. This is all morphological data, so they had to track the daughters and the old poles and the new poles and then score it manually. So this was really painstaking work, and they credit the two students who really did a lion's share of this data collection, and those are the authors, Audrey Pernecka, who's a PhD student from Brazil, and Christian Butz, an undergraduate who did that work.
So you had a conversation with the senior author, didn't you, Camilla?
Yes, I was able to have an email exchange. She is a native of Sweden, And that's where she got her PhD at the University of Gothenburg, studying microbiology and molecular biology, in particular looking at gut bacteria, their growth rate, do they transfer genes in vivo, et cetera. To do her work, she actually commuted between her home university, Denmark's Technical University, and Uppsala University, and Karolinska Institute in Sweden, because the expertise that she needed was spread out among those three different universities. But she reflects saying, I was young, I was carefree, I could travel and live in a suitcase. It was a very exciting time. She felt lucky to build her network and friendships with these amazing, smart, and kind researchers. During that period, she also met her future husband, who was a professor in the U.S. at the time, and that motivated her to pursue postdoctoral training in the U.S. So she moved to Baltimore and joined the Center of Vaccine Development at the University of Maryland School of Medicine, and worked on a project to develop a vaccine for Vibrio cholera, which of course causes the terrible disease that can be pandemic, cholera. So their strategy was, perhaps we can identify a key protective antigen by identifying proteins that are only expressed in vivo in the gut, but not in the lab. So they set up a GFP promoter screen to look for genes that are specifically turned on when Vibrio cholera binds to human epithelial cells. But around the same time, her family was growing. Her husband got a faculty position at UCSD. She was in Maryland. She convinced her PI in Maryland to let her continue the project, but to do the work at UCSD. So that worked out great. They were very happy, wonderful place to raise their son. Unfortunately, the cholera project did not work out so well. It was high risk. She was working in isolation. So eventually she decided, and together decided, it was time to put that project to rest. And she now joined her husband's research group, which focuses on evolution of viruses and bacteria. So her husband is the senior author on this project, so they have been collaborators for some time. Mm-hmm. Outside the lab, she enjoys knitting and genealogy. And in particular, she joined MyHeritage, a web-based genealogy site, and was proud to discover that she is a great-great-great-great-great-grandchild of a famous Viking, Ragnar Lothbrok. So she's was happy to discover that through her love of genealogy.
She doesn't look like a Viking. No, maybe she does.
Her other hobby is, of course, her son. She is proud that he's now a 21-year-old junior at UC Berkeley, and he's majoring in biology, but is interested in larger organisms, not microorganisms. So he is right now pursuing his interest in conservation biology, and she's quite proud of it and happy for him. Her advice for younger scientists is that there's not just one thing in science that you can pursue, so don't fret over finding the exact one path for your professional life. If you like biology, just grab something and run with it. She's also noticed that American-trained students that she's met think that they need to come up with a huge discovery all by themselves during their PhD. But she said it just doesn't work that way. Don't put that kind of pressure on yourself. Instead, take advantage of what the lab is doing, what your PI knows well. It's a great training environment, and in time, you will find your own path and begin to generate your own ideas. But don't think that at the beginning of your research career, you're going to come up with some brilliant idea that none of the great minds who've come before you had not already thought of. Be humble, she says.
Boy, that's great advice. That's really good advice.
Yeah. And again, she wanted to give a lot of credit to the her co-authors on this paper who did the painstaking, elegant work of tracking where these micelles form and the growth advantage that the daughters that have arisen from a mother cell who spawned a micelle.
Cool.
But again, I want to point out that they do a really beautiful job of having color schematics that partner with all their phase images of bacteria and also their fluorescent images of bacteria. She also wanted to thank a colleague in France who provided them with a key strain. So this is Ariel Linder. He provided them with a strain that tagged a green fluorescent protein onto another bacterial protein that was known to accumulate at the poles. So that allowed them to treat their bacteria with streptomycin, and then were amazed to see that this marker for poles actually did accumulate in the poles and bud off in these minicells. So they have a lovely image of that experiment, figure 5. And she said that was just, they were ecstatic in the lab the day that they were able to use the strain from their colleague, Ariel Linder, to do that experiment. So again, showing that science is very collaborative.
Nice. Thank you, Michele.
Yeah.
And thank you, Elio.
My pleasure.
All right, let me read a few email here before we wrap up. Neeraj writes, "Hi, TWiM Manisomes. It was great listening to the latest TWiM, and just when I thought I was already amazed to saturation by the diverse world of fungi, thanks for the explanation, Elio, along came Salmonella Bon Jovi, and the title, It's My Life, totally suits it. I was particularly intrigued by the ability of the pathogen to downregulate the flagellar machinery, but was left wondering if this is probably one of the reasons why flagellin, a component protein of flagella, has never been developed as a vaccine target against gram-negative bacteria. Kindly update if you think or know otherwise." And Michael knows otherwise, right?
Jerry Peer and his colleagues have been evaluating whether or not flagella and flagellin from Pseudomonas aeruginosa is useful in vaccines, and I'll put the link to the paper into the show notes.
Okay. And just when I was thinking that maybe the same is true for LPSO antigen, along came Elio again and asked my question. What a psychic. It was like I was part of a classroom. It was like I was part of a classroom with 50 million questions in my head, but now I can only remember 2 of those. I'm sure they will come back to haunt me later when I hit the bed, but it's the kind of mental exercise that I absolutely thrive on, and that's why there's nothing more fascinating to me than the mysteriously complex world of the microbes. It's like anything we can innately and adaptively to try and curb their invasion. The microbes have an alternative mechanism right up their sleeve. So please continue the great work and continue to educate us. Happy holidays, Neeraj. P.S. Thinking of gram-negatives. Another question, something to do with the usage of gram-negative E. coli for making proteins. I was wondering if we know the precise reason for the preferential use of gram-negative E. coli for heterologous expression of protein. Was it simply because it was the first model system ever used, or was there a more rational scientific reasoning behind it? In today's world, several gram-positive like Lactococcus lactis, Bacillus subtilis, etc., are also being used for protein production, but E. coli remains the favorite model organism. A literature search didn't yield much clarity, so I thought I would relay my queries to the esteemed panel of TWiM.
You can find this answer in the back of your favorite molecular biology catalog, where it lists the strains of bacteria for cloning. And most of the strains that are optimal for cloning have their restriction and modification systems deleted. So you can put in foreign plasmids that won't get restricted by the restriction and modification systems of the bacterium. And so you can get heterologous gene expression. So it all gets reduced to a one-word answer, and it was because of the genetics that was available to the E. coli biologists at the time molecular biology was being founded.
But theoretically, the same could be done with Bacillus subtilis, yes?
Oh, yeah, absolutely.
And it is.
And it is.
So it's just,
that's his point.
It's widely used.
But
so it's just historical.
Didn't Joshua Lederberg say, here's E. coli, let's use it?
Yeah.
Pretty much. Well, he was lucky. I mean, he used a strain that was amazingly, coincidentally happy to mate.
Yeah, yeah.
But I think the reason is really historical. It was the first bug that about which we could manipulate, like Michael has said. So that became the standard.
Okay. All right, there's a letter from Lisa. Dear TWiM team, this past semester I designed and taught my first ever graduate course on bacterial pathogenesis at San Jose State University. As an avid and longtime listener to TWiM and having done both my graduate and postdoctoral training in virology, the thought of delving into bacterial pathogenesis was daunting, to say the least. Where to start? So I began my TWiM listening, and it was wonderful. In particular, having the opportunity to listen into discussions by experts in the field on current and fundamental topics in microbiology was absolutely essential. The papers discussed on TWiM provided critical insight into microbiology topics that I could incorporate into my lectures: CRISPR-Cas9, antibiotic resistance, harnessing bacteriophages for antimicrobial therapy, V. cholerae, type 6 secretion, etc. I included links to relevant episodes during lecture, and many of my students began listening and commenting on how wonderful it was to have access to these episodes as a resource for their learning. Beyond scientific content, the career advice was also incredibly helpful. For example, a number of the students were interested in the clinical laboratory scientist program, and I suggested listening to TWiM 52 to gain further insight into the program. Please accept my thanks to the entire TWiM team for all the time and effort that goes into these podcasts. They have had a huge positive impact in my little corner of the world. On a separate note, I ran into Vincent at ASV this past summer, and he encouraged me to not give up on my dream of becoming professor. 27 faculty applications later, I recently accepted a tenure-track assistant professor position at Midwestern University, and I am so excited to start. There are not words to express how grateful I am for all of the podcasts and words of encouragements over the years. These podcasts truly bring science to life. Thank you. Lisa Kronstadt is currently a postdoc in Stanford University. Well, that's great.
Wow. She's now an assistant professor.
I guess she has, at the time of writing, she was still at Stanford.
She just accepted.
Yeah, what a wonderful letter.
I might print this out and put it on my refrigerator at home. It's really lovely. Thank you, Lisa.
All right, one is from Johannes. Dear TWiM team, I stumbled on a really non-scientific website over a highly interesting article in Science News, which led me to an article in Cell, Open Access. And this is an article that Michael did for us a few episodes ago: Emergence and Spread of Basal Lineages of Yersinia pestis During the Neolithic Decline. Remember that, Michael?
Yes.
And so Johannes suggests that we do the paper, but of course, Johannes, we've already done it, because he says, I leave the discussion of the article to you. Your entertaining way of breaking down the paper might help to debunk the reaction it had on this website. So this is a website which the article is entitled, So This Is How Every Zombie Starts. So
what?
Yeah, right. Plague DNA is no apocalypse. Thank you for your tireless work to educate the world. Johannes is from Leiden, the Netherlands. And one more here. Rich writes, guys, first of all, many thanks for another very interesting podcast. Rich writes about the Yersinia paper. SNVs were mentioned, single nucleotide variants, when you discussed the Yersinia paper, which made me think of SNPs, single nucleotide polymorphisms, and I wasn't clear what the difference was. I carried out an internet search, and I believe that SNP and SNV can be used interchangeably, but if a distinction is made, then a SNV is generally used to refer to an individual, while a SNP refers to a SNV that is present in multiple individuals in a population over 1%. I would be interested to know if this is the rule or whether there is something missing.
That's a taxonomy question for the special taxonomy people.
I think that's correct, though.
Yeah, it is.
I think the SNVs are,
And with the case of
rare.
this plague paper, we didn't have enough of frozen people to effectively
make it
or
a
enough
SNP.
frozen teeth to figure out the story.
Right. And one last from Steve, who writes, I know you don't have listener picks on TWiM, but if you did, this would be mine. And this is an article on Gizmodo and a video showing the death of a single-celled organism. And it's called Contemplate Existence as you watch the mesmerizing death of a single-celled organism. So someone made a video of a Blepharisma dying. Thank you, Steve. All right, that's it for TWiM 194. You can find it wherever you play your podcasts. You have a podcast player on your phone or your tablet. You can listen. And if you do, please subscribe. It helps us to know how many people who are listening. If you like what we do, please consider supporting us financially. There are a couple of ways you can do that over at microbe.tv/contribute. And, of course, questions and comments, TWiM@microbe.tv. Today on TWiM, Michele Swanson from the University of Michigan. Thank you, Michele.
Thank you. And by the way, are we getting some good responses from our listeners? I think you'd put out a poll.
I did, yes. I think we have a good response, but we'd like more. Thank you for reminding me.
Yeah.
You can go over to asm.org/TWiM poll. asm.org/TWiM poll. One word, TWIMPOLL. 12 questions, 5 minutes. We'd just like to know more about you, our listeners.
Really looking forward to that.
We have some. We have done the same on TWiV, and we have interesting findings there, but we'd like to know about our TWiM listeners as well. Yes. Elio Schaechter is at Small Things Considered. Thank you, Elio.
I think my microphone is acting up.
Okay.
But thank you anyhow.
Thank you.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.